Welcome back. In the second part of Lecture 15, we're going to continue talking about the kidneys and the urinary system. We're going to talk about nephrons a little bit more, a little about something called the JG, or juxtaglomerular apparatus, and then we'll start talking about urine formation, and we'll finish that in Part 3. This material is from Chapter 25 in Merib. Let's first talk a little bit more about the nephron and the different parts of the nephron, since this is so important to urine formation. Here you can see in the boxed area, someplace where we've already been, we've already seen the afferent arteriole, which is here. We've looked at the efferent arteriole, and of course the glomerulus itself, which is the tuft of capillaries that's inside the glomerular capsule that we looked at before. Remember, this is also called Bowman's capsule. So we have the afferent arteriole bringing blood into the glomerulus, goes through the glomerulus, and then comes out the efferent arteriole. The efferent arteriole then will go form the peritubular capillaries, and will also form, in those certain cases, the vasa recta when we're talking about juxtamedullary type nephrons. Now, what we want to do is continue our discussion of the nephron and talk about the different parts that it has and how these parts are constructed. First of all, you'll notice that the tube that's directly connected to the glomerular capsule is known as the proximal convoluted tubule. We abbreviate that as PCT, proximal convoluted tubule. It's called proximal because it's the one that's closest to the glomerular capsule, as you see here, and it's called convoluted because of the winding, twisting manner in which it goes through the cortex of the kidney. Now, the proximal convoluted tubule, as you see on the right, has cells that are lined with a brush border. That is, these have microvilli. And as we talked about before, the proximal convoluted tubule cells are very suited to absorption of material from the fluid that's inside the tubule. The next portion of the nephron is known as the nephron loop, or sometimes called the loop of Henle. You'll notice this comes from the proximal convoluted tubule, goes down into the medulla, turns around, and comes back up toward the cortex. And you'll notice that the nephron loop is a thin segment. The thin segment of the descending nephron loop is lined with simple squamous epithelium. And as we've talked about several times in the course and in A&P1, simple squamous epithelium is very good at allowing substances to diffuse across the cell layer. In fact, as we'll see later on, this simple squamous epithelium is very good at allowing water to escape from inside the tubules and move out this way. Following the thin descending limb of the nephron loop, we have this segment that begins right here. This is the thick ascending nephron loop that's once again lined with cuboidal or low columnar cells. These cells are very good at secretion and absorption. And following this, then, we have the distal convoluted tubule, which is the last segment of the nephron. We abbreviate this DCT. Once again, this is called distal because it's further away in absolute distance from the glomerular capsule. In other words, if we were to take this nephron and straighten it out, we'd see that the distal convoluted tubule is much further away from the glomerular capsule. We call it convoluted once again because the tubules twist and wind throughout the cortex of the kidney. One thing you'll notice, though, is that the distal convoluted tubule loops back, and part of it is located right between the afferent arteriole and the efferent arteriole. So this area right in here is what's known as the juxtaglomerular apparatus. Juxta once again meaning next to, glomerular meaning glomerulus. We're going to talk about that in the next slide. The distal convoluted tubule itself is lined with simple cuboidal cells. As we mentioned before when we looked at the light micrograph, these cells do not have any microvilli. The order of the parts of the nephron is important to know. Once again, we start at the glomerular capsule. We go to the proximal convoluted tubule, the thin descending loop, the thick ascending loop. Then we go to the distal convoluted tubule, and the distal convoluted tubule is going to go into structures that we talked about before that are known as collecting ducts. So the collecting ducts or collecting tubules are those things that we saw running down through the renal pyramid that terminated at the renal papilla and dripped urine into the minor calyx. Now let's turn our attention to the juxtaglomerular apparatus. As I mentioned just a few seconds ago, this is a combination of a distal convoluted tubule sandwiched in between the afferent arteriole and the efferent arteriole, as you see here. So this area right in here is known as the juxtaglomerular apparatus. Sometimes we refer to this as the JG apparatus. And if you notice at the center of this, right in this small triangular area here, we have a set of cells that are known as juxtaglomerular cells. In fact, these are the cells that will secrete renin. Remember, we talked about renin earlier when we talked about the endocrine system and said that renin is secreted whenever the blood pressure goes down, or whenever the kidney senses that we have lower concentrations of sodium in the plasma. So there are sensing mechanisms that will tell their juxtaglomerular cells to secrete renin, and that in turn will bring blood pressure back up and will cause reabsorption of sodium and water at the kidney. Now you'll notice another area in here that's known as the macula densa, right in here. These cells of the distal convoluted tubule you'll see are slightly different than the other cells of the distal convoluted tubule. In fact, these cells are osmoreceptors. In other words, these will detect the osmolarity of the fluid that's passing through this tubule. If they detect a lower concentration of sodium in the tubular fluid, what they'll do is instruct the JG cells to secrete renin, and that in turn will enhance sodium absorption at the kidney and restore the normal levels of sodium in the body. So these juxtaglomerular cells, the macula densa, and also the afferent and efferent arteriolar muscle cells all function in sensing blood pressure and in sensing concentrations of sodium. So the juxtaglomerular apparatus is a combination of the macula densa cells plus the JG cells or juxtaglomerular cells. This is what the juxtaglomerular apparatus actually is. Now we're going to begin talking about glomerular filtration and urine formation in a little bit. Before we do that, however, I just wanted to compare glomerular filtrate and urine and look at how these two things compare. First of all, before we start looking at the table, take a look at how we measure concentrations of different solutes and electrolytes. In the top panel, you notice we measure these in terms of something that's called milliequivalents per liter. And as you'll see down here in this definition, milliequivalents is nothing more than a measure of concentration. So the higher the number of milliequivalents, the higher the concentration of solute in a particular fluid. The reason we use milliequivalents is because you'll notice that all of these solutes that we're talking about over here have some type of charge, whether it's positive or negative. These are all charged moieties. So we use concentrations in terms of milliequivalents per liter because this is easier to relate positive and negative charges and see how they balance. Now if you look at the bottom of the table, you'll notice that we measure concentrations of other solutes in units of milligrams per 100 milliliters. And if you remember from what we talked about before, 100 milliliters can also be expressed as a deciliter. So this is milligrams per 100 milliliters or milligrams per deciliter. And what we're doing is measuring the concentration of solutes like we see in the bottom here, like glucose, urea, uric acid, creatinine. These are non-charge type solutes. When we have that, we measure these in milligrams per deciliter. Okay, so let's take a look at our first comparison, and that is between plasma and glomerular filtrate, these two boxes that we see here. Now, if you take a look at this, you'll notice that the concentrations of all the different solutes that we have on the left are pretty much identical between the plasma and the glomerular filtrate. We really don't see a difference at all. In fact, the glomerular filtrate, that fluid that enters the capsule that comes from the plasma, is pretty much identical with the plasma. The only major difference is that it doesn't have a high concentration of proteins in it. Now, if we compare the glomerular filtrate to the urine, which are these second two boxes, you'll notice how different the concentrations of solute are. And there are a couple of things going on that we're going to introduce in a little while called tubular reabsorption and tubular secretion. So we won't discuss those in depth here, but all we'll do right now is take a look at the concentrations of these things and talk about what's happening just in general terms. Notice, for example, that sodium in the glomerular filtrate is present at 142 milliequivalents per liter. However, in the urine, this drops to 128 milliequivalents per liter. What this means is that there was some sodium that was in the filtrate and then in the tubes of the nephron that had been taken out of that and put back into the blood. We'll see later on that the process of taking things out of the tubules of the nephron, bringing them back into the blood, is known as reabsorption. On the other hand, take a look at something like urea down in the bottom portion of the table here and notice that its concentration in the glomerular filtrate is 26 milligrams per deciliter. However, in the urine, its concentration is 1820. Well, that seems kind of weird. How can we get more stuff in the urine than what we had in the filtrate, which was the beginning of urine in the first place? Well, it turns out that there's another process called tubular secretion. And what's happening here is we're taking things from the blood and we're actually pulling those into the tubular fluid to make the urine. So this is a way for the body to get rid of more things than actually what appear in the filtrate. And that's a process called tubular secretion that we're going to talk about in a little while. We can also see, for example, tubular secretion here with chloride. We can also see it with potassium. So there are several different solutes that we have this process of tubular secretion going on, and you can also see some tubular reabsorption. One of the primary things I just want to mention before we leave this is take a look at the levels of glucose in the glomerular filtrate versus the urine. You'll notice that we start out with 100 milligrams per deciliter in the filtrate, whereas we wind up usually with 0 milligrams per deciliter in the urine. What's happening here is the process of tubular reabsorption, and once again what that means is that we're taking the glucose from the tubules of the nephron, taking it out and bringing it back into the blood that will go back into the body. So we're reclaiming all of the glucose that's in the filtrate and moving it back to the body so that we don't lose it. So the text on the bottom rehashes just what we said. The glomerular filtrate is about the same composition as plasma. The major difference is that we normally don't have a high level of protein present in the filtrate. However, notice once again that the glomerular filtrate and the urine are very, very different. And these are the processes that we're going to come back to and talk about a little later. How does the urine become very different from the filtrate? Now, next we want to talk just a little bit about urine formation. We say that about 125 milliliters per minute of the total 1200 milliliters per minute of blood that passes through the glomerulus becomes filtrate. What does that mean? Remember that what we're doing at the kidney is the plasma from the capillaries in the glomerulus is being squeezed out into the capsule, and that accumulates about 125 milliliters of fluid from both kidneys in all nephrons every minute. And that equates to about 180 liters a day. In the very first slides of the urinary system lectures, we mentioned that we couldn't possibly excrete 180 liters a day, or we'd be dead in a matter of minutes. So something has to happen to this 180 liters that's filtrate to reclaim most of that fluid, and as we said before, we reclaim about 99% of all this fluid, so what we excrete as urine is around 1.8 liters on average every day. Now, glomerular filtration is a process that adds to the volume of urine produced. This should be obvious to you by now. We've talked about this several times. Remember, glomerular filtration is moving the fluid in the plasma into the glomerular capsule, and this is going to then flow through the nephron. So we've got to do this in order to produce urine. So this is the very important first step. So substances from here move from the blood into the glomerular capsule. This is the process of glomerular filtration. Now, one of the things we talked about just a couple of seconds ago was about tubular reabsorption. It's important that you remember the definition of this. This is moving substances from the renal tubules into the blood of the peritubular capillaries, and eventually this blood is going to be returned to the body. If you take a look at the left-hand side of this panel over here on the lower right, you'll see that tubular reabsorption is taking place right here. We're taking fluid that went into this tube, pulling it out and putting it back in the peritubular capillaries, which is eventually going to go back to the body. So very important things that we don't want to get rid of undergo tubular reabsorption. Since we're pulling fluid out of the tubules here and moving it back into the body, this is actually going to reduce the volume of urine that we produce because it's taking fluid out. Now we have an alternative process that I mentioned a couple of minutes ago. This is called tubular secretion. This is the opposite of tubular reabsorption. So this is actually going to add to the volume of urine, and you can see tubular secretion illustrated on the right side. Notice that what we're doing now is taking substances that are in the blood of the peritubular capillaries and moving these into the tubules. This will eventually become urine. Okay, so this is eventually going to be excreted from the body. So this is the way that we looked at before, where we get more of substances in the tubular fluid than we had in the filtrate to begin with. Now, the other point I'll make is that we can algebraically look at how urine is formed when we consider that both glomerular filtration and tubular secretion add to the volume of urine produced. Tubular reabsorption subtracts from it. So these are two that add to the volume of urine produced. This is the one that subtracts from it. Now, the last thing we want to talk about in this part is about glomerular filtration. We're going to talk a little bit more detail about the forces involved in glomerular filtration and how the process actually occurs. What we want to talk about here, though, is the very first step, and in particular, we want to talk about the anatomy that accomplishes the glomerular filtration. Recalling what we talked about before, we have an afferent arteriole, an efferent arteriole, and in between, of course, we have the glomerulus. Recall that the afferent arteriole is bigger in diameter than the efferent. This causes a high pressure in the glomerular capillaries, which we're going to come back to in a little while. The process of filtration remember, is moving plasma through the filtration slits of the podocytes and into the capsule that we have right here, right? Bowman's capsule or the glomerular capsule. This is going to be the very beginnings of urine. When fluid comes out of here and goes into the capsule, we now call it, remember, filtrate. Okay, so the filtrate is the very, very beginnings of urine. Now let's take a close-up look at some of these glomerular capillaries and the structures that are associated with them. What we're going to talk about is something that's known as the filtration membrane. And the filtration membrane, as you see here, consists of three different things. First of all, the fenestrated vessels of the glomerular capillaries. Notice the capillary endothelial cells have holes in them. These are fenestrations, or holes. Remember we said that there were three different types of capillaries: continuous, fenestrated, and sinusoidal. This is an example of the second type of capillary. So what the fenestrations allow is larger molecules to escape from these capillaries than would otherwise happen in, for example, other areas of the body. The second layer of the filtration membrane is the basement membrane. Remember, all epithelium has a basement membrane associated with it. These capillaries are no different. So there's a thin basement membrane outside the capillary endothelial cells, and the basement membrane is the same basement membrane that we had, for example, below the skin, below any other epithelium in the body. And finally, the third layer of the filtration membrane consists of the filtration slits of the pedicels and the podocytes. Remember, these cells basically have interdigitating processes that come out like this, and they go in between one another like that and form these filtration slits. And so these are the slit pores, and you can see some of these illustrated in the diagram up here. The spaces that we have between the epithelium of the cells are the filtration slits. Now, in order for material to make it out into the capsule, as we mentioned before, that material has to be small enough to get through the fenestrations in the capillary, the basement membrane, and also the filtration slits that we have in the podocytes that surround these capillaries. So the combination of these three things is known as the filtration membrane of the kidney. And there are many pathological states that the filtration membrane actually changes and will change the properties of the filtration at the kidney. So this is important for you to understand. So when we talk about glomerular filtration, we could say that it's primarily a mechanical process based primarily on molecular size. In other words, which molecules are small enough to fit through each of these three layers of the filtration membrane. However, that's really not the total story. The filtration membrane also does have a charge component to it. That is, there is an electrical charge that regulates the things that are filtered as well. And that charge primarily comes from the basement membrane. The basement membrane consists of negatively charged polysaccharides and those sort of things that actually repels negatively charged proteins from within the capillaries. So there is a charge component to filtration. However, we're going to consider that the main filtration is based on mechanical processes, primarily based on molecular size. Okay, so I'll see you in part 3, and we'll begin talking a little bit more about glomerular filtration, some of the forces involved, and the beginnings of urine formation. See you then.